Good morning or good afternoon, everyone, depending on where you are joining us from, and welcome to today's webinar. My name is Eliza Barry from Business Review, and I will be your host for today. It is our pleasure to have BioPharmaSpec with us today, who will be presenting today's webinar titled Host Cell Protein Analysis by Mass Spectrometry. Today's guest speakers are Christina Morris, Senior Scientist, and Gillian Roma, Senior Scientist at BioPharmaSpec. I'd like to welcome you to our platform on 24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you receive via email to rejoin the session. In order to ask questions, you can send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click Submit. We will allocate some time at the end of the session to address any questions or thoughts that you may have. Please use the yellow Help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now, please allow me to welcome Gillian.
Thanks, Eliza. Thank you, everybody, for joining us today. During this webinar, we're going to discuss some of the advantages and disadvantages of ELISA analysis for the detection of host cell proteins, and how LC-MS can be used as a complementary orthogonal approach to ELISA. We'll also discuss our workflow for LC-MS analysis of host cell proteins. And at the end, we'll summarize everything we discussed today and have a short Q&A session. So first, I just want to give you a little background about our company. BioPharmaSpec is a privately owned company that was founded by Professor Howard Morris and Dr. Andrew Reason in 2014. We are a contract research organization that focuses on the structural, physiochemical, and biophysical characterization of biopharmaceutical products, primarily using state-of-the-art mass spec and chromatography techniques. BioPharmaSpec currently has 2 labs, one in the UK and one in the United States. These are just some of the services we offer and some of the previously characterized products we've done. Many of the services we provide were pioneered by our scientists over the last 30 years. BioPharmaSpec has extensive experience in the characterization of many products, including monoclonal antibodies, fusion proteins, and heavily glycosylated molecules. Our management team was also involved in the initial characterization of many of these innovator products and are now able to assist biosimilar companies in the development of their products. Next, I just want to give some background information on host cell proteins, about what they are and why they're important to us. So host cell proteins are recombinant proteins that are produced in host cells. These host cells are commonly of mammalian, yeast, or bacterial origin. Purification steps throughout the manufacturing products are intended to remove most of the contaminants. However, at the end of the process, some contaminants may still remain. So these host cell protein contaminants may lead to reduced drug efficacy or unintended immunogenic responses. So this figure summarizes the entire manufacturing process of a therapeutic protein. Host cell protein analysis generally focuses on the last 3 steps of this figure. The purification step itself is a multi-step process that should be monitored for the clearance of host cell proteins by demonstrating the removal of proteins throughout the purification process. And after the purification process, there should be less than 100 parts per million of host cell protein. Host cell proteins frequently co-purify with the biopharmaceutical product. And evidence of this co-purification can be observed in the traditional ELISA by a nonlinear sample dilution. So in occasions like this, when nonlinear sample dilutions are observed, other orthogonal methods should be looked at to investigate these occurrences. An example of co-purification is the phospholipase B-like-2, PLBL2, which has been found to co-purify in multiple CHO-derived products. So this is a common impurity in products throughout the biopharmaceutical industry. It's important to note that not all commercially available ELISA kits are able to detect this protein. ELISA is the traditional method for host cell protein analysis. Some of the advantages of ELISA include good reproducibility, high sensitivity, and high throughput potential. It is estimated, though, that ELISA can only detect about 70% of host cell proteins that are potentially in a product. Some of the advantages of using a mass spec-based orthogonal technique is that we have the advantage of identifying a wider population of host cell proteins in a de novo manner. Mass spectrometry provides identification and quantitation of specific host cell proteins in the product, and it's not limited to host cell proteins that produce an immunogenic response. LC-MS is very sensitive and able to detect host cell proteins at sub-10 part per million amounts, and frequently it's being requested by the regulators as an orthogonal approach. So LC-MS is not a replacement for the ELISA tests, but it's a good complementary method. So for the next portion of our presentation, Christina Morris is going to compare some of the attributes of the ELISA method to our mass spec method.
Thank you, Gillian. Now I'll take you through a few tables outlining further some of the differences between the two techniques to highlight why we think LC-MS is a powerful tool for HCP analysis. So we have here specifications of sensitivity and dynamic range. ELISAs have traditionally been able to offer very high selectivity and sensitivity for antigens at particularly low levels in a mixture of other molecules. But LC-MS is also capable of measuring down to femtomole amounts of material with very high precision and confidence. The dynamic range of LC-MS is also 4, sometimes 5 orders of magnitude, which means we can detect very low levels of contaminants among very high levels of drug product within a single run and without the need to perform serial dilutions. Moving on, other assay considerations include the method development time and the scope of compounds or HCPs that can be identified by these different approaches. In comparison to the short method development time of LC-MS, the production of anti-HCP antibodies for ELISAs is often time-consuming, and because they are multi-step processes, as you can see in the image below, that rely on biological molecules, the lot-to-lot variation needs to be carefully monitored. Another feature is that HCP-ELISAs don't currently offer HCP-specific identification. Instead, they are measured as a population where it's not possible to distinguish any HCPs of particular interest. And very importantly, as Gillian has mentioned, if an HCP cannot evoke an immunological response to raise antibodies against, then it will not be detected by the antibody-based ELISA. LC-MS instead is capable of detecting individual HCPs regardless of their immunogenicity. And then lastly, we have a comparison of expertise or training between these two methods and considerations regarding method process flexibility that we'd like to highlight. Often the training requirements for the basic operation of immunoassays are lower and tend to be shorter than LC-MS learning, although the evolution of software and equipment has helped to simplify the running of these instruments and certain elements of data interpretation. It's critical in ELISA to have good specificity between the antibody and HCP antigen to reduce the chances of cross-reactivity or false positives and low sensitivity. In cases where this needs to be readdressed, however, the demands on cost and time are higher than any LC-MS method changes. And I think it's time now for a poll question, so I'll hand over to Eliza.
Thank you so much. Okay, everybody, so it is now time for the first polling question of today, and the question is, are you currently using or do you plan to use MS analysis for HCP identification in your products? And the options that we have today are, yes, we are using a combination of MS analysis and another orthogonal method for HCP identification, such as ELISA. We also have the option, no, we will only use a generic ELISA kit for HCP identification. We also have, no, we will use only use custom-developed ELISAs and or ELISA kits for HCP identification. And the final option we have is we are not currently using MS analysis for HCP for analysis, but we are considering implementing this as an orthogonal technique. If everybody would like to make their selection, and we'll just give you a minute or so to select the answer most applicable to yourself, and then we will move on to reveal the results. Just a reminder to the audience, if you do have any questions in the meantime, if you would like to submit them in via the questions widget, you just type them into the box at the top left-hand corner of your screen and click submit. And we will address any questions or thoughts that you have about the webinar in the Q&A section towards the end. Okay, so I think everybody has probably made their selection now, so let's reveal the results. Okay, interesting. So 37.5% of you voted for yes, we are using a combination of MS analysis and another orthogonal method for HCP identification, such as ELISA. 12.5% voted for no, we will only use a generic ELISA kit for HCP identification. 6.3% voted for no, we will only use custom-developed ELISAs and or ELISA kits for HCP identification, and the overwhelming majority, 43.8%, voted for we are not currently using MS analysis for HCP for analysis, but we are considering implementing this as an orthogonal technique. So what do you think about those results? Are they surprising, Christina and Gillian? What do you think?
I'd say that, yeah, it's not hugely surprising. I guess ELISA is the gold standard, so given that it's the traditional method of choice, those two options aren't hugely a surprise, but I think we'll start to see movement hopefully towards using LC-MS as the community investigates the merits. So anyone that's in the middle two categories will take an interest, and this presentation will help to develop that.
Great, thank you. Okay, I will now pass it back to you. Thank you, everybody, for voting.
Thanks, Eliza. So I'm going to be talking about the next part of our presentation, which is going to discuss some of the regulatory guidelines. Since these can be a bit long-winded, I'm just going to discuss some of the key points of each regulation, and at the end, I'll summarize everything that we think is important to know about this analysis. So for the EMA, they discuss the monitoring of host cell proteins as a quality attribute that should be monitored with appropriate controls. And then they emphasize that controls that are appropriate for one product or process may not always be appropriate for the next product. So these controls really need to be product specific and looked at pretty frequently. The FDA, the U.S. regulatory body, recognizes the information outlined in the ICH Q6B as their current thinking on the topic of host cell protein characterization. So in the Q6B, they discuss that criteria for impurities is required. Host cell protein is defined as an impurity. They say that impurities should be minimized throughout the manufacturing process. They also say that impurities should be screened using a sensitive ELISA, and impurities should be screened using a sensitive assay, and ELISA isn't specified as the only assay that you can use. In the ICH Q5E, they discuss that previously qualified host cell protein assays may no longer be appropriate if there's been a change in the manufacturing process, and the assay should be requalified. And finally, in the ICH Q7, they discuss that removal of host cell proteins should be demonstrated, and testing should be performed throughout the manufacturing process to show an active decrease in the host cell protein levels. So just to summarize, it is the responsibility of the product manufacturer to determine which host cell proteins to monitor as their critical quality attribute. and also to determine which analytical method best suits them. There aren't specific defined safety guidelines in any of the regulations for host cell protein amounts. These safety limits are defined by the regulator and the manufacturer on a product-specific level. But typically, we look for a detection limit of 1 to 100 parts per million. And then finally, it's important to keep monitoring your host cell proteins throughout your purification process to make sure your clearance levels are actively decreasing by the end of your final drug product. Next, we're going to talk about peptide mapping using LC-MS. So this is a really important method that we use for our host cell protein analysis. Peptide mapping is used to characterize a protein by determining its peptide mass fingerprint. Peptide mapping allows for the identification by mass and characterization of the MS-MS of these masses by inducing fragmentation to determine sequences. Peptide mapping is really important for host cell protein analysis because it allows us to assign a specific retention time and fragmentation pattern to a known host cell protein peptide. And then we will screen all of our subsequent samples for these known peptides. Modern mass spectrometers, such as a QTOF type instrument that we use here, are capable of generating fragment ions from these peptides at the same time as generation of intact mass peptide data. The fragmentation ions obtained are used to confirm the identity of the peptide, again, through the generation of amino acid sequence information, which can then be used for either database interrogation or protein identification in the cases of protein mixtures or unknown proteins. Or we can compare back to the expected sequence of a known protein. So in this figure, our general workflow is we receive the protein, we do a reduction and alkylation experiment, we do a digest with a known protease, and then we put it on the instrument for LC-MS analysis. So I just want to introduce some of the vocabulary that we use in case you're not really familiar with mass spec analysis. So mass spectrometry is an analytical technique which identifies proteins based on the mass of the sample. And then we couple it with a liquid chromatography system which will separate the sample based on the stationary phases of choice. There's many stationary phases you can choose from, but two of the most popular ones are reversed phase separation or size exclusion separation. Most of our host cell protein analysis uses the reverse phase separation technique. So this figure shows what we call a total ion current. So this is the sum of all of the ions we observed during the LC-MS analysis. The x-axis is time, and the y-axis is ion intensity. So if we wanted to screen our total ion current for one specific mass, we would produce something called an extracted ion chromatogram. So an extracted ion chromatogram is created by searching for one specific peptide with a known mass-to-charge ratio. So in this case, we know that this is a known protein, and we know the enzyme we use to digest it. Therefore, we can determine the approximate size of all of the peptides present in the sample. So when we search for one specific peptide, we call this an extracted ion chromatogram, which is the top figure you see here. The next thing we're going to discuss are the two common acquisition methods used for peptide mapping. They're also very important for host cell protein analysis. The first one is called information-dependent acquisition. So for this method, the instrument cycles between a survey scan and a targeted MS-MS fragmentation scan of a single precursor ion. So this is also called DDA, or data-dependent acquisition, and these terms change depending on the manufacturer of your mass spec. So in this figure, our top box shows what we call the survey scan, and in this there are several signals. So for information-dependent acquisition, the highest level signals are chosen to be further fragmented. So in this case, the yellow precursor is chosen for further fragmentation, and so is the green, and so is the red. The nice thing about this method is all of the MS-MS fragmentation is attributable to one known precursor, and it makes the interpretation much more straightforward. However, one of the downsides is that it doesn't choose the low-level signals for fragmentation. So in contrast to the other method of acquisition, so this is data-independent acquisition, which can also be called MSE or SWAT, depending on, again, what model of mass spec you're using. So for this, the instrument cycles between MS survey scans and MS-MS fragmentation scans, but these are independent of the mass-to-charge signals identified in the survey scan. MS-MS fragmentation spectra can be derived from several precursors, making it difficult to determine which fragmentation data is attributable to which single precursor. So in this example we have here, we have the same survey scan for the top, but at the bottom we only have one MS-MS scan. So all of these signals have been fragmented together. The yellow, green, and red signals, but also some of the lower level black signals have also been fragmented. So in this, it's difficult to determine which fragmentation came from which initial precursor. So this is just some of the, a comparison of both methods. So if we're looking at sensitivity, the data-independent method is more sensitive because it's not just looking at the most intense signals, it's looking at all of them. For reproducibility, again, the data-independent method is more reproducible than the data-dependent method. For data-dependent methods, the signal that's chosen for fragmentation may change from time to time, while if you're fragmenting everything, you'll get roughly the same spectra each time. Now, for assignment ambiguity, the data-dependent method is superior because fragmentation is attributable to one defined precursor signal. So this is important for our host cell protein analysis, especially for our library building step, which we will discuss further in a few slides. So next, I want to talk about our MS-MS spectra. So this is the fragmentation that I've been talking about. So for this, some, or in this case, all mass-to-charge ratios observed in the mass spec spectrum can be further fragmented with MS-MS. And from this, we can determine specific amino acid sequences of the peptide. To do this, we use a mixture of what we call B and Y ions. So B ions are fragmented from the C to the N terminus of the peptide, and Y ions are in the reverse direction, N terminus to C terminus. So this is an example of MSE data. So this is a data-independent method, and it shows some of the labeled Y ions that are visible in the spectra. So with this, we know the predicted sequence, so we are able to go through and label each of these Y ions that we see. We start at the lower end of the spectra. We can see that the series starts at this 478 mass. And then you can see with the red boxes, it moves further up the range. And we're able to determine that the spaces in between the masses correspond to known masses of amino acids. So we're able to determine that the sequence of this peptide is histidine, tyrosine, leucine or isoleucine, aspartic acid, alanine, and leucine or isoleucine. It's important to note that leucine and isoleucine have the same mass. Therefore, it can't be differentiated by mass spec alone. Other techniques, such as gas phase sequencing, need to be used to definitively determine leucine and isoleucine. And I think it is time for another poll question.
Thank you so much. Yes, it is now time for the second polling question of today. And the second question today is, do you use CRO support for HCP analysis? And the options that we have today is, yes, we make extensive use of CROs as we do not have in-house analytics. We also have the option of, yes, we use CROs for most work, but also have a low level of in-house analytics. Or we have, yes, we use CROs for some work, but also have significant in-house analytics. Or the final option we have is, we do not use CROs as we perform all analyses in-house. So if everybody would like to make their selection now, which is most applicable to them, and I'll just give you the reminder that if you do have any questions, to submit them, if you submit them into the box at the top left-hand corner of your screen and click submit, and we will be addressing any questions that you have had throughout the webinar in the Q&A section towards the end. Okay, I'll just give everyone a couple more seconds to vote, and then we will reveal the answers. Okay, let's reveal the results. Okay, interesting. So 12.5% voted for yes, we make extensive use of CROs as we do have the in-house analytics. 31.3% voted for yes, we use CROs for most work, but also have a low level of in-house analytics. And 31.3% also voted for yes, we use CROs for some work, but also have significant in-house analytics. And 25% voted for we do not use CROs as we perform all analyses in-house. Okay, what do you think of those results?
Well, so I would suspect that most companies are still doing their host cell protein analysis using ELISA. And that's a pretty common thing to have in your in-house analytics. But I would think that as regulators are asking for more orthogonal methods to support the ELISA, we might start seeing a trend of using CROs for this orthogonal testing.
That's great. Thank you so much. I'll pass it over to you now. Thank you.
Thank you. So now we are going to discuss using LCMS as our orthogonal method and how the workflow looks, which is quite a bit different from the traditional ELISA method. So we use a four-step process. The first step is the ion library creation. So for this step, we use the whole cell extract, or the lysate, and we will create an ion library using our peptide mapping technique, where we map the retention times and BNY ions of our potential host cell proteins in our lysate. And this will create a large database that we will screen against throughout the subsequent samples that we're going to look at. For step two, we do a qualitative identification. So we use a highly sensitive instrument combined with data-independent acquisition to screen mid-process samples and final drug product samples against our original ion lysate library. I should say the original library is screened against a database. Typically, a SwissProt database is used. This is a high-quality, annotated, and non-redundant protein sequence database that we download, and it'll be specific to the species used to create your drug product. So the third step is a relative quantitation step. So with this, we spike in known internal spikes, and we use this to monitor the changes in host cell protein amounts throughout the purification process. After we've completed the qualitative identification and the relative quantitation, we'll choose ideal proteins or peptides to move on to our next step, which is absolute quantitation. For the absolute quantitation, we use heavy labeled peptides to quantitate using a multiple reaction monitoring technique. So we'll discuss all of these in the next few slides. So for us, steps 1 through 3 are performed with a SciEx triple top 6600, which is a very sensitive mass spectrometer. And our step 4 is performed with a triple quad mass spec, which provides very accurate quantitation. The biopharma specs approach is to use a SciEx workflow with swath acquisition for our host cell protein analysis. The swath acquisition is very powerful because it's extremely sensitive. Swath uses mass isolation windows stepped across a mass range to cover a mass to charge distribution of peptides. So just to show what our capabilities are, we have performed a case study using some known standards and also using our SciEx instrument with our SciEx software. So for this study, we obtained a human K562 lysate to replicate a whole cell lysate that could be used for a biopharmaceutical product. And we've spiked this with our relative quantitation standards. We've gone through our peptide mapping technique of reduction, alkylation, and digestion, and acquisition using our data-dependent methods. The next sample that we looked at was a dynamic range standard that has multiple proteins at various levels. So we used this as a mid-purification process sample. So the point is that this shows all of the peptides that we would expect to see in our host cell as a host cell protein in high levels, but without the final drug product. And we analyzed this using our data-independent method, in addition to our data-dependent method. To replicate a final drug product sample, we used a NISTMAB reference standard, and then we spiked in very low levels of our dynamic range standard, along with our relative quantitation standards. And again, this simulated drug product was reduced, alkylated, and analyzed by LC-MS, also using our data-independent method. So for this final product, we screened this data against the peptide map that we created with the original host cell protein cell extract, our K562 sample. So first, we're going to talk about our library creation. This is probably the most important step of our workflow. So again, this uses our peptide mapping technique to map the proteolytically digested proteins to specific retention times matched with B and Y ions that we see. The fragment ions, the B and Y ions, are used to determine the identity of the peptides and then identify the proteins from which they were derived. So for this method, we use a data-dependent method exclusively because, as we talked about previously, this reduces the ambiguity of the assigned peptides. So after we've created our library, we run a statistical analysis on this using some of the bioinformatics tools that SciEx has. In this case, we use SciEx Protein Pilot software. So it's important to do the statistical analysis to assess the accuracy of your method. To do this, we calculate something called a false discovery rate. So this is a method that's used to determine the number of false positives in a data set. So our false discovery rate is calculated by looking at the number of matches in a decoy database comprised of reverse peptide sequences. So in this case, we've run it through the SDR analysis, and it has told us that it thinks that 1,863 proteins can be identified out of our lysate. So if we compare this to the certificate of analysis for our lysate standard that we purchased, our certificate of analysis recommends that one should be able to identify 1,802 proteins. So we can determine that our method has successfully identified the correct amount of proteins, maybe even a little bit more. And these are high confidence identifications. So after we run it through the software, we're able to run our subsequent samples. So our NIST-MAB sample that has been spiked with this dynamic range standard. And the results of our case study showed that we were able to detect down to a 5 parts per million level using our data-independent method. So in this case, we collected our data-independent method data and screened it against the data-dependent data. So we matched against retention times and B and Y ions. So it's important to know that the data-independent method was able to detect down to a lower level than the data-dependent method. And this is because of some of the things we looked at earlier that compared both of these methods. So it's generally a more sensitive method. Also using the swath analysis is able to increase our sensitivity. It's also important to note that there was one protein that was identified by the data-dependent method and not by the data-independent method. And that's why it's important to run both methods, just to make sure you're getting a complete picture. This is an example of some of the software that we use to look at the detection of our low-level contaminants. So this uses our spectral screening method. So the top figure is the total ion current of the NIST-MAB sample with our dynamic range spike in it. The middle figure is the extracted ion chromatogram of the specific myoglobin peptide that we've been looking at. The myoglobin peptide was the 5 parts per million low-level contaminant. So if you compare the extracted ion chromatogram, which it looks like our peak is coming out at about 28 minutes, to the figure above at the total ion current, you'll see that this myoglobin peak isn't initially visible in the total ion current because it's being overshadowed by the background of the high levels of drug product, or the NIST-MAB in this case. But if we run it through our software and we know to look for this specific peptide at this retention time and which B and Y ions to look for, we can easily find this very low-level peptide. And we can see that there are several very strong Y ions visible in the bottom picture. So this all confirms this all confirms that we know the identity of this peptide. So after we've done our identification, we can go back and do our relative quantitation. So for this, we'll initially assess the linearity of our internal protein spikes across a large range. In this case, we did 20 parts per million to 1,000 parts per million. We'll spike in 2 or 3 levels of various levels and quantitate each host cell protein against our spiked-in level. We use the high-3 method of quantitation at this stage in the workflow. The high-3 method uses the average integrated signal intensity of the top 3 most intense peptides. And this signal is generally proportional to the absolute amount of protein in the sample. It's important to note that differences in ionization efficiency between the spiked peptides and the host cell proteins may cause some inaccuracy of the calculated host cell protein amount. However, since the quantitation approach is the same across multiple samples throughout the purification process, we use this method to assess change in the host cell protein levels across the different preparations. And if there is a measurable increase or decrease in levels in host cell proteins, we can assess this at different points. So just to summarize our quantitation method for relative quantitation at this stage, we use internal protein spikes to quantitate our contaminants. Again, the estimates will be compromised due to the differences in ionization efficiencies. After this stage, there are more accurate mass spec-based methods to use to quantitate, and these will provide a more accurate number for host cell protein amount. However, relative quantitation is very useful in monitoring the clearance levels of host cell proteins throughout the manufacturing and purification process. And then, again, at the end of the process, we would expect to see less than 100 parts per million. So for the next part of our presentation, Christina is going to discuss our absolute quantitation method.
Great, thanks. So I'll give a brief summary of what absolute quantitation is and how and why we perform it. In comparison to relative quantitation, absolute quantitation makes use of stable, heavy labeled isotope standards, which reduces the inherent differences in ionization efficiency between different peptides. Typically, instruments like Orbitraps or triple quadrupole mass spectrometers are well suited and designed to this technique. So in the schematic below and in the following slides, we focus on a triple quadrupole instrument method. So firstly, the instrument selects precursor ions belonging to the peptide of interest that are from a predetermined list that we input into the method. This takes place in mass filter quadrupole 1. Fragmentation of the precursor ion is then induced in the Q2 collision cell, generating the resultant product ions in the third quadrupole, Q3. Each Q1 to Q3 transition from precursor to product ion constitutes a single reaction monitoring event, and what we want to do is select strong transitions for the best sensitivity. When multiple product ions are followed, this is termed multiple reaction monitoring. In the diagram, we have an example of this. So there are 3 Y product ions that have been monitored and used to identify and quantify a particular peptide, which is also distinguished by its reintervention time. So to aid MS detection, proteins are digested into peptides, which Gillian has already covered, and they act as models of the intact HCP protein. It follows, therefore, that the amount detected of each peptide is theoretically equivalent to the amount of each protein. In cases where 2 or more peptides are quantified per HCP, we take an average of this response. It's important to note at this stage that peptides from these proteins are selected according to certain criteria, including charge state, sequence length, and composition. So we aim to avoid residues that can be readily oxidized and deamidated and therefore interfere with detection. Once those candidate precursors have been chosen and peptide identification confirmed using instruments with higher resolving power, so for example the QTOF, synthetically stable isotopically labeled standards are requested by us for manufacture, and these are spiked into a sample at known amounts. So the HCP peptide concentration is then calculated by comparing the intensities of the heavy labeled internal standard with the native peptide equivalent. As an example data set in this next slide, we show in the top trace a native peptide signal, and the bottom corresponds to the labeled version of this peptide. To the right of each trace are these single reaction monitoring events we spoke about, and you have, for example, the transition from precursor ion, so the 686.36, to the product ion 574.33 in the native trace. Commonly, stable isotope-labeled peptides contain amino acids with carbon-13 or nitrogen-15 to generate a predefined mass shift, in this case plus 10 Daltons. You'll note, though, that the precursor ion shift between traces is plus 5 and not plus 10 Daltons, and that's because we measure mass over charge, and these are doubly charged signals. Finally, to develop a robust method that is accurate over the concentration range for which proteins are under investigation, we create calibration curves for our standard peptides. In cases where there may be interference, for example, sample loss from non-specific hydrophobic interactions that give rise to nonlinear behavior, then we'll select different candidate peptides for a protein. And so for this reason, it's important to have several candidates per host cell protein. So to summarize then, the regulators are interested in seeing evidence that can corroborate HCP-ELISA results, and LC-MS has proven it can provide a complementary alternative that is accurate and sensitive with high reproducibility. In our case study, we've shown how HCP identification begins by developing the reference ion library using the originator cell line of the drug product and information-dependent MS acquisitions. And then using data-independent swath acquisitions, we can reproducibly search for the low-level HCP signals down to 5 to 10 ppm. Finally, high-throughput quantitation can be done using label-free methods, such as spiking in protein internal standards and measuring their peptide signal areas from extracted ion chromatogram traces. Or for several HCP of interest, absolute quantitation can be performed with heavy isotope label standards and targeted data-dependent acquisition like multiple reaction monitoring, which if required can measure down to single-digit ppm levels. So thank you for listening, and please see our details below for any further questions or feedback after the webinar. But I will ask the host now if we have time for a few questions.
Thank you so much. Okay, yes, it is now time for the Q&A section. We've received quite a lot of questions in. So I'm going to kick things off with the first question of today, which is, when should I be implementing HCP-MS phase 3 or earlier?
I'll take this question. So we recommend to start early in the development process because you'd want to show that as you develop your product and your purification protocols that all of the host cell proteins are being cleared. So all of the ones that are immunogenic and not. So if you use LC-MS to screen from very early on, you're minimizing your risk of carryover of those potentially harmful host cell proteins.
That's great. Thank you so much. Okay, and the second question of today is, can I replace one of my ELISA methods, i.e. the kit or the custom-developed ELISA, with a mass spec-based method?
So in our opinion, MS is used as an orthogonal approach, i.e. to be done in tandem with the HCP-ELISA screening. So in order to provide the broadest degree of coverage of HCPs within a product, we'd recommend using both methods, and that way you get the best knowledge of your product. But ultimately, advice from the regulators should also be sought when making those decisions, I would say.
Thank you, that's great. Okay, and the next question we have today is, how long does a 2D LC library creation take? Can you discuss how this would work?
Yeah, so, well, the answer to that is it's really product specific. A more complicated host cell lysate will take longer to characterize. So I guess the advantages of the 2D LC method is that you get much better separation, and therefore you'll improve the depth of your reference library that you're using for your screening. But it does take a lot more time. It can take several more weeks, maybe a month. Kind of hard to give a definitive answer without knowing what product we're discussing.
That's great. Thank you so much. Okay, and the next question we have is, how many HCPs do you generally see in a final drug product?
So again, this is another product-specific question. Typically, when we move from the mid-stage, we see somewhere in the hundreds, and then by the final stage, somewhere in the range of 8 to 10 host cell proteins. And we think this is pretty accurate. This is what we've seen in a lot of published case studies. So yeah, generally 8 to 10.
Great, thank you. Okay, I think we've got time for one final question then. And the last question of today will be, okay, does a product-specific cell lysate need to be used for every project, or can a previously characterized library be used? Do you have a previously created generalized library to use?
I can take that, Gillian. So I would say every cell system used to create the drug product will have a unique protein landscape. So while it's possible to build on previous libraries, we'd recommend characterizing the cell originator that's been used in the manufacturing process, because different growth or stress conditions can have an impact on the proteome that is expressed.
That's great, thank you. Okay, so unfortunately, that is all we're going to have time for today regarding all of your questions, but I'm sure we will get back to you in the meantime. So thank you everybody who submitted and interacted with us in the polling questions today. So that just leaves me to thank Christina and Gillian for what I'm sure we all agree was a great presentation, and to BioPharmaSpec for sponsoring this session. To the attendees, you receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. We look forward to sharing further webinars with you, so please do keep an eye out on our website and follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group, Business Review Webinars. So thank you everybody for joining once again, and I hope you all have a lovely day.